Uh, Continuing the theme of neurorestorative type treatment strategies, it's my pleasure to introduce our next speaker, Dr. John McDonald from the Washington University School of Medicine. Dr. McDonald will be telling us about embryonal stem cells and their ability to promote functional recovery in spinal injured animals. Dr. McDonald.
Well, thank you for that introduction. I'd like to thank the organizers for the invitation, and I'd really like to thank Jeff for that really fantastic talk and great introduction to what I'm going to tell you next. But really, both of the work that we're doing in both of our labs is very, very similar. So first, I'd like to just start off, give you a little background about where I come from and what the center that I run, what the goal is. And really, you know, we do a lot of basic science, and I'm going to tell you about some of our work, but in addition, a lot of the advances and concepts of regeneration that I think are important for the educated lay-level audience to understand. But in addition, I'm also very interested in clinical sciences. I'm a neurologist by training as well. And so I'll also present today some advances in concepts of treatment, because there are a lot of things that can be done now. And we'll also, just like Jeff mentioned, discuss some of the upcoming treatments that will be likely in the future. But more importantly, you know, I think what the design of our center and what my interest is really the intersection between both of these. So it's the intersection between basic and clinical sciences. So what can we offer, you know, as advances now? And there's a considerable number of things. So this is a kind of epitomizes this concept. This was a new rehabilitation facility that was just completed on the WashU campus. So it's on the rest of the campus. And what it really integrates is the spinal cord injury labs are on the 4th floor. So there we're doing animal research and culture work. But the patient work, the inpatient spinal cord injury neurorehabilitative unit is 1 floor below. So it's really one of the first facilities to integrate both of those in the same place. And I think that's what the goal of our center and the goal of what my interests are is really looking at the intersection of basic science and clinical sciences and determining what can be translated into new human treatments. So this is really the overview of the program. Really, we have everything under one roof, so it's very unusual for one center to have everything. So we have the acute spinal cord injury team, which we call the STATeam, a neuro-rehab spinal cord injury unit, outpatient spinal cord injury program, links to a lot of external resources which are so important to people with spinal cord injury, and the spinal cord injury international level research program. It's called the Restorative Treatment and Research Program. And we have this now both for adults and pediatrics. This is the spinal trauma assessment and treatment team called the STATeam. And really, as you all know, I mean, transverse myelitis is a spinal cord injury. It's probably one of the most complex medical surgical disorders and requires a lot of integrated care from many different services. So these are really the eight clinical services required for the clinical management of acute spinal cord injury. And so we really formed a team so that the entire team sees the patient from day one all the way through rehabilitation. And really, this really increases the efficiency of the treatment, dramatically reduces length of stay, reduces costs, and reduces complications. So how does all this relate to transverse myelitis? Well, you know, most of the numbers that are quoted for spinal cord injury refer to traumatic injuries. And, but there's many more numbers of non-traumatic causes of spinal cord injury, and transverse myelitis is just one of those. So this is just a rough depiction of the non-traumatic causes of spinal cord injury, and this varies dramatically depending on whether you're in a city in the country or what country you're in. But of the demyelinative disorders, transverse myelitis is included there. Other infectious disorders include polio, post-polio syndrome, degenerative disorders including ALS, tumors, orthopedic conditions, ischemia including AVMs. In fact, a subset of transverse myelitis is probably misdiagnosed as AVMs. And then congenital disorders all fall into this category. And so our center focuses on treatment of all these, including the traumatic causes of spinal cord injury. So transverse myelitis, as you've heard over the last couple of days, really encompasses many different etiologies. But in my thinking, at least as a scientist and approach from a regenerative standpoint, which I'm going to focus on today, there's really two useful groupings. One is there's a focal selective white matter injury. And this is very similar to what Jeff just showed you, where they inject the ethidium bromide and you get pretty much selective loss of the myelinating cells and some astrocytes, but largely many of the axons are preserved. And then there's a second group where you get really widespread necrosis involving gray matter. And essentially you end up with a lesion almost identical to traumatic spinal cord injury. And what I want to tell you, get across to you is that most of the mechanisms that are involved in the progression of the injury, whether it's caused by trauma, an inflammatory condition, or stroke, the progression of secondary injury and those mechanisms that are involved are largely similar from all the etiologies. And the approach to repair of the nervous system is very similar. So all the things we learn from different neurological disorders as far as limiting secondary injury and promoting regeneration are going to be applicable to other disorders in the nervous system as well. So what treatments do we have to offer already for people with spinal cord injury? So we have medical surgical care, stabilization for trauma, methylprednisolone and steroids, as you've heard a lot about here. Of course, for spinal cord injury, traumatically, this is the only FDA-approved treatment. Standard rehabilitation, functional electrical stimulation has an important role. There's many advanced FES treatments that are now FDA-approved, including the VoCare system and freehand system that allows someone to regain control over their bladder, regain use of a paralyzed hand. Those have been FDA-approved since 1997 and 1998. Functional tendon transfers is a reversible treatment that is often overlooked that can allow someone to regain use of a single hand. Partial weight supported walking, activity-dependent rehab programs, advanced assistive programs, and participation in clinical trials are other things that need to be offered to patients with spinal cord injury. Well, this is the problem, spinal cord injury. We've heard a lot about this. In trauma, it's broken fragments of bone and soft ligaments that impinge on the very soft spinal cord, about the consistency of jello. And that initial injury, again, whether it's trauma or initiated by lack of blood flow, rapidly enlarges over minutes to hours to involve a large part of the spinal cord, and you're often left with a cyst or a hole in the middle of the spinal cord. So this would be consistent with a more severe form of transverse myelitis. And the reason this occurs is because of the vascular blood supply to the cord. When the cord swells, like if someone were to punch in the eye, it rapidly swells inside a fixed canal. Once the pressure overcomes venous blood flow pressure, which is low, about 40 to 50 millimeters of mercury, you end up with a venous infarct, and you largely lose the inside of the cord. But remembering how the cord is organized, the inside is mainly made up of gray matter, and you're always left with some variable donut-like rim of tissue that's primarily white matter around that. And as Jeff alluded to, it's white matter that ends up being most important in spinal cord injury. For example, the segmental loss of gray matter at C4 is not very important. You'll have someone who has, preserved arm, leg, and bladder and bowel function. However, if you damage white matter at the same level, you have someone who's tetraplegic and has no control over bowel and bladder. So this is years after an injury. This is the spinal cord. These are the vertebral bodies. This is anterior, this is posterior. Here's the spinal cord. Here's that cyst. So it's a fluid-filled cyst at the site where the trauma originally occurred, between C4 and C5. And there's a variable rim of tissue around that. If you cut a cross-section here, it looks like this. This outer continuous line outlines the outside of the spinal cord. The inner dotted line outlines the cyst. So here's that donut-like rim of tissue that's primarily made up of white matter. And of course, MRI and the studies we can use today don't demonstrate functional connections. And that's why Jeff was pointing out the importance of doing physiology to demonstrate that there's really functional connections through there. But in most cases, there are connections that go across the lesion, many of which aren't functional because of segmental demyelination, as Jeff pointed out in his talk. But what this really illustrates is that, and what I want to point out, is that the goal is only partial repair, that severe damage, even like this, can still leave considerable function. In fact, this is someone who can compete in a triathlon. So this is someone who can walk, run, and do a number of different things. They're not normal, but they can have considerable function. So first, before we get to regeneration, I think there's a couple of important new concepts in neural injury that you need to understand. And Jeff alluded to a number of these. So apoptosis plays an important role in addition to necrosis. Oligodendrocytes are highly vulnerable to release of glutamate that occurs from damaged axons. And as Jeff pointed out, each oligodendrocyte reaches out and myelinates 10 to 20 different axons. So loss of a single oligodendrocyte can produce exponential loss of function. In addition to the early cell death that I pointed out in that earlier slide, there's a second wave of delayed oligodendrocyte apoptosis or programmed cell death that occurs for weeks to up to a month in rodents in the degenerating white matter tracts. And this probably contributes to substantial loss of function, although that has not been proven yet. And of course, methylprednisolone interacts and tries to limit this progression of secondary injury. So this slide just really quickly overviews, I think, some of the important concepts that have been borne out over the last 10 years in terms of what's needed for regeneration. First of all, spontaneous regeneration is the rule rather than the exception. Some spontaneous regeneration occurs in everyone. And the goal should be improvement in quality of life. A cure is not required. The goal is partial repair. And I think that first slide really illustrated this. And that you can get to disproportional functional gains for small anatomical improvements. And in the final, there won't be any one magic bullet, but that multi-stage interventions will be required, each providing incremental benefits. And as Jeff pointed out, we need to use clinically relevant, doable animal models. And of the spontaneous regeneration that does occur, this is limited by inhibitory factors that actually inhibit some of that spontaneous regeneration. And there's probably hundreds of these. And what's interesting to know is that just masking single proteins can produce substantial improvements in spontaneous regeneration. That is, we don't need to know all the answers. And the nervous system is not static. So the nervous system is constantly turning over. So it's very different from what we used to think. There's stem cells, as Jeff pointed out, that are endogenous, that are capable of making new cells all the way throughout life. We're starting to understand those endogenous stem cells through transplantation, and we'll talk about and focus on this today. And I also want to point out the importance of pattern neural activity. That is, during development, pattern neural activity is essential for normal development to occur. And normal development in the nervous system is probably our best model of regeneration. So therefore, pattern neural activity will probably be very important to optimize regeneration following a nervous system injury. And I'll allude a little bit to that at the end. So I just want to emphasize once again that a cure is not required. The goal is partial repair. Here's that variable donut-like rim of tissue. And this really points out that most of the important clinical tracts that produce function between the brain and the body are located in this outer rim. So I think it's easy to understand how it'll be easier to prevent the progression of secondary injury than it will be to repair the nervous system once it's already occurred. However, once the injury is already over, the only thing we can do is try to promote regeneration. And of all the potential strategies, and these are just some of the most promising ones, you can list these as far as their level of likely difficulty. Which are likely to occur first, which are going to be the most difficult to do. And this is my view of this, the level of difficulty with the most difficult up here. This is the red zone, and what I mean by the red zone is something that's never been achieved in any animal model. So we haven't been able to get long-track axonal regrowth and appropriate reconnection. So even if we can get the axons down, we don't have any reasonable idea that we can get them to make the right connections. We're getting better at getting cells to survive and promote axonal outgrowth. Neuronal cell birth is something that we understand can occur from endogenous stem cells. Using genetics, can get cellular molecule expression, axonal sprouting. But as Jeff pointed out, remyelination is one of the more doable things. We know that dysmyelination in particular is something that contributes to dysfunction after nervous system injury. And this does not require a formation of new circuits. In some cases, glial cell birth, like the astrocytosis that occurs in the scar at the level of the injury, is something we actually want to try to stop. So there's some things that occur with regeneration that are inappropriate. And some of the pain that develops after spinal cord injury is an example of inappropriate regrowth. So what I'd like to move on to now is really focus on just one form of regeneration, which is cellular replacement. And I want to just focus on one example of one source of cells. Jeff covered a number of great examples, and I want to look at the earliest form of stem cell called an embryonic stem cell. First, I'd just like to point out that these are very complex problems, and the problem of repair and regeneration is constantly changing as a function of time after the injury. So some treatments may be very effective only if given very early, like the red line would be methylprednisolone. Other things that may prevent, for example, that delayed death of oligodendrocytes may be effective in a subchronic time, and transplantation may be effective in a long-term time point. So these are important things to think about in the animal models that we use. So what are ES cells? I think you've heard a lot about this in the news, and as scientists, I don't think we've done a good job of describing what they are. Embryonic stem cells are capable of undergoing indefinite replication. That is, they don't age when they divide. They're pluripotent, meaning they can make every cell in the body. They can contribute to every tissue in the body. However, they're not totipotent. They can't make a new organism by themselves. They have the best proof of genetic normality. That is, we can take an embryonic stem cell, put it back into an early blastocyst, and have the genetics of that cell contribute to the organism. But most importantly, they're highly genetically manipulable. So you've probably heard a lot about transgenic animals and how much that's changed our ability to study in neurosciences. Well, now we have that same ability in single ES cells. So each gene has 2 copies or 2 alleles, and we can do double gene knockouts and knock-ins now in single cells. And before ES cells, that wasn't possible. So this is a little video that goes through how embryonic stem cells are made. It's primarily done through in vitro fertilization. It's been done in most species, including mouse, rat, and human. And basically you take sperm and egg and produce fertilization. Now if we grow that fertilized egg in a dish over a couple of days, you get a small group of cells, and this is called a blastocyst. If we divide that in half, this is the inner cell mass. These are embryonic stem cells. So we now know enough to be able to take those out by a procedure called immunodissection and grow them in a culture dish. And as you'll see, that really does provide an indefinite source. The cells divide every 10 to 12 hours. If we begin the week with 1 flask of 5 million cells, we'll end the week with 256 flasks. So it really, of the ES cells, for example, in the case of humans that have already been isolated, in theory that's enough to populate all the laboratories of the world. So because ES cells can make any form of cell, they can make appropriate tissue, but in an inappropriate place. So for example, if we want to look at nervous system transplantation, you don't want lung being made in the brain. So the best way to limit that is to try to restrict the cells prior to transplantation. So that's exactly what we do. We use a procedure that was developed by a colleague at WashU, and we can take the embryonic stem cells, grow them floating in a dish. It really resembles that early blastocyst as an embryoid body. Then expose them in the last 4 days to retinoic acid. This is the same stimulus that causes the brain basically to form in the embryo. And at the end of 8 days, the majority of these cells are neural precursor cells that are capable of forming the 3 main types of cells in the nervous system: neurons, astrocytes, and oligodendrocytes. We've also developed another procedure where we can rapidly produce precursors for oligodendrocytes, what we call an oligosphere. So we do this to help restrict the cells prior to transplantation so that they don't form other tissues in the brain. If we look at this, this is a view of one of these embryoid bodies using scanning electron microscopy. These are individual cells. This is a much higher power view using transmission electron microscopy. And inside, you can actually see what look like neural tubes. And the cells, as they divide, they pull down like they do in the normal neural tube and divide. If you look using immunocytochemistry, I won't go through the details, there's very few differentiated cells. The majority of the cells are nestin positive. This is a marker of an early neural progenitor, what we call a tripotential progenitor, capable of making astrocytes, oligos, and neurons. So we can take those embryoid bodies, grind them up, put them in culture, show that we can form neurons, 2 different types of astrocytes, and the typical oligodendrocyte with its many processes. We can also grow neuronal cultures. These are cultures of neurons with bundles of axons. So these are the same kinds of connections that we need to try to repair in the spinal cord. And we can show that we can form many types of different types of neurons, many of which are found in the normal spinal cord. We can also show that they form connections between each other. Basically, these are individual cells. This is the nucleus, the cell body. These are the processes. These little red dots are markers of the presynaptic component of a synapse, or a connection between a neuron. So you can see really hundreds of connections on this single cell. And we can use EM to show that these are really truly synapses. I'll just walk you through this. This is the postsynaptic cell. This is the presynaptic component. This is the density that you typically see at the synapse. These are the synaptic vesicles. So these little vesicles fuse and release the neurotransmitter that act on receptors here. So these are round vesicles, typical of excitatory neurotransmitter. We can also show inhibitory synapses. This is one that's typical. Again, this is the postsynaptic cell, presynaptic. This is the density. And these are elongated as opposed to round vesicles, more typical of an inhibitory neurotransmitter. Our colleagues Jim Huttner has also shown that these cells are physiologic, that they produce the typical inhibitory and excitatory postsynaptic potentials. These are electrophysiologic indices of connections between cells. So now I want to, using that same paradigm, we've looked at transplantation into the contusion injury model. This is a model that Jeff briefly outlined. It really simulates a lot of the pathology that occurs in the human condition. This is the basic model. This is a rat, and this is what we used in this condition. We do a laminectomy, and we basically stabilize the spine and drop a tiny little weight on the open spinal cord. And this really simulates the type of injury found in a human spinal cord injury. This is the experimental timeline. We did the contusion injury, and we did a pretty severe one, such that the animals, when they recovered, weren't able to walk. We transplanted 9 days after transplantation. The reason we chose this time point is we clearly wanted to be out past the time point where most of the cell death was already over. That is, so that we focus on regeneration. And furthermore, most of the bad inflammatory cytokines that can kill cells and could kill the transplanted cells are going away by this time point. And then we analyzed 2 and 5 weeks later and also assessed the ability of the animals to walk. We used multiple stem cell lines, and that's important because you may have cell line specific effects. And as Jeff pointed out, it's very important to use multiple markers to accurately label these cells and identify the cells as transplanted cells. We've also genetically engineered the cells so that they basically glow green. So you can see this is an individual cell 2 weeks after transplantation in the injured cord, and these are all the little processes. So really hundreds of cells. And here at higher power you can see individual processes. So using the genetic potential of these cells has made it very easy to be able to track these cells and see where the connections go and what they connect to. This is really a schematic diagram 2 weeks after transplantation of the relative distribution of the cells. This is that central gray matter. This is the outer white matter. And this is a longitudinal section. This is where the contusion injury was done. So here's the typical cyst or syrinx that occurs. Basically, this becomes filled up with the transplanted cells, but the cells rapidly migrate, just as Jeff showed in his demyelination models, well over a centimeter in both directions in the cord. We can count these and show that they distribute like that. They basically fill up the cavity. They form cells that look like neurons. So this is an individual cell. This is the nucleus that fits inside that cell. And these are the processes surrounding the lesion. We can show, using a bunch of immunolabeling, and I won't go through the details, that we can show all the 3 main types of cells. And if we count these, the majority of the cells turned into oligodendrocytes, about 60%. Less than 10% turned into neurons, and about the balance turned into astrocytes. And in these studies, we specifically didn't look at the Schwann cells like Jeff showed in his study, but that's something that would be very interesting for us to go back and look at. But more importantly, we didn't see any abnormal tissue formation. So it appears that that early restriction we did was sufficient to prevent these cells from forming normal tissue, but in the wrong place, something called a teratoma. What was surprising, we saw some behavioral recovery. We didn't expect to see this in the nascent cells. This is this behavioral scale that goes from 0 all the way up to 21, so this is a broken scale here. This is the days after injury. And you can see the control group spontaneously recovers just as humans do after an injury, and then they plateau. And the key is they plateau at around 8 to 8.5. And this is an animal that can't walk. In contrast, animals that received the transplant after about 3 weeks showed statistical differences. And with most behavioral scales, this is a very small difference. It's only 2 points. But as you'll see, I'll show you examples of the average of each. So an animal that's 8 and an animal that's 10. And you'll see that even my grandmother would be able to tell the difference between an animal that can walk and can't walk. So we've repeated this multiple times now. And we've added additional controls and see the same consistent differences. We've also carried it out to longer time points, and it persists. But this is an animal from the control group 6 weeks after transplantation. It's a BBB score of 8, so it's an average from that group. And what you'll see, whoops, you'll see that this animal can move, but it won't be able to lift its tail. It can move its hind limbs. I don't know why this is not working. So this, let's go back. So here we go. He wasn't motivated. So here he can move his hind limbs, but he can't bear weight, and he drags his tail. This is the level of the injury. They're like that some days. So this is a littermate control, again 6 weeks after transplant. Now this animal received embryonic stem cells. You can see that it can bear weight, it can lift its tail, it can even stand on its hind limbs. But the walking's far from normal. You know, I mean, this walking is about like the walking of the guy that ran in that triathlon. But you know, for that person to finish a triathlon was a big deal. Anyhow, those were very early studies, and we're really starting to try to build on those. And our initial hunch, just as Jeff pointed out, is that because the behavior occurred fairly quickly, the majority of the cells turned into oligodendrocytes, and the majority of the cells that migrated a long distance were oligodendrocytes. We suspected that it was myelination that may be what's responsible for that behavioral recovery. But those studies didn't prove that. And so that's what we're trying to do right now. So we started to investigate in more detail how well oligodendrocytes derived from ES cells can actually myelinate, using similar studies to what Jeff pointed out in the last talk. So using that oligosphere protocol, we can get cultures that have well over 90% oligodendrocytes. So this is a single oligodendrocyte with all the branches. So we can rapidly enrich for these cells. We can show in culture, this is a culture, a mixed culture, derived from ES cells. This is a single oligodendrocyte with all its branches. And it's overlaid over a phase image, so you can see axons. These are axons, shown here with the white line. So a single oligodendrocyte will reach out and wrap multiple segments of different axons, just as they should in vivo. And we can use EM to show the early forms of myelin. So this occurs within 9 days. So it's incredibly rapid, much more rapid than oligodendrocytes derived from the primary or embryonic brain. Just as Jeff has shown, we used myelin deficient models as well as ethidium bromide demyelination models to prove that in the adult nervous system, in the injured nervous system, that these cells are capable of myelinating. And basically, this is a low-power view. In this case, it's a shiver mouse. This is a mouse that cannot make myelin basic protein. It's an integral protein in myelin. So what's neat after transplantation, if you just do anti-MBP immunoreactivity, this represents all the transplanted cells. But you can see distance is a limitation, so the cells only migrate a limited distance. You can show at much higher power that represents myelin. Again, this is myelin basic protein immunoreactivity. This is a single cell. This is a nucleus that fits inside that. It's reaching out and myelinating axon here with multiple branches. At a lower power view, you can see this is in a ESL transplanted group. See lots of green indicating myelin and white matter, and this is the sham control. At higher power for here, you can see these are individual axons, these spaces, and this is typical of the immunoreactivity wrapping around a single axon. And we can use EM to show that this really represents myelin. This is an axon. This is the mitochondria inside the axon, and this is early compact myelin. But you can still see immature forms with the inner and outer tongues, typical of the wrapping myelin. This is a picture where we did DAB immunoreactivity for the mouse transplanted cells. This is in the same model that Jeff showed, using ethidium bromide to demyelinate in the rat dorsal column, and we can show that the ES cells can myelinate there as well. We've also used other techniques to label the cells. So in this case, I mean, it'd be really nice to be able to label cells in a living animal and then track them over time. So what we did here, working with Jeff Bolte at NIH, was to label the cells before transplantation with a paramagnetic marker. So then we can use MRI to track the transplanted cells. So we used the same contusion injury model and then transplanted. This is 2 weeks after transplantation. This is the spinal cord. This is the gray matter. What's gray is actually white matter. And the black signal are the transplanted cells. And you can see that they migrate a long distance, just as we showed in the previous studies. So hopefully, when we get to human studies, we'll be able to track the cells in the patient, because of course we don't have the option to take it in the spinal cord. This is a three-dimensional view of that same animal. So why don't we stop there? I'd like to thank all the people in my laboratory that have contributed to this work, are really a great number of people over about 5 years, and all my collaborators at WashU and NIH, in particular Dennis Choi, David Gottlieb, David Gutman, Chung Shu, Mark Jacquin, Gene Johnson, and Carl Larayson. And then all the people that care for these spinal cord injured animals, because as all you know, these animals have the same problems humans do. They have bladder and bowel trouble. They need therapy. You know, they need to be taken care of 7 days a week, 3 times a day, including holidays. So without them, none of these studies would be possible. And I'd just like to finish with another note that it's important to stay active. And this is not a joke. We're actually doing this in the lab, not in this way. But that activity is very important, even in a paralyzed person. And hopefully later this year we'll be able to share some work with you proving that. But just keep that in mind. Thanks.
Any questions for Dr. McDonald?
Read that there are currently 6 embryonic stem cell colonies.
Yeah, so with she's referring to human embryonic stem cell lines that have been isolated basically through in vitro fertilization. And in theory, 6 would be enough, you know. But because of genetic variability and problems that could occur with the lines, you're going to want the ability to isolate additional ones. But if we could only use those 6, even that would be great, you know. But you know, currently, you know, I think the decision that we're trying to go through in our country on whether or not to use human stem cells, you know, is really unrelated to the pro-life issue. The pro-life issue is already something that's been decided. You know, this is completely irrelevant, really, compared to that issue. And you know, there's so much potential behind these stem cells, and I think some of our work that Jeff and others have shown, you know, demonstrates the potential of some of these cells and really the power behind them from genetics. And it'd be a real shame if our government doesn't stand behind that and provide funding. Yeah, there. Sure. Yeah. Is there an active lobbying group to support federal funding for stem cell research in humans? And the answer is yes. There's many groups. Unfortunately, that got started awfully late. And but they're very active now, and hopefully they'll be able to win that over. Yeah, in fact, one of the best places is just to contact the Christopher Reeve Paralysis Foundation, okay? And they're available on the website, and they're incredibly active and can link you to all the other sites that are involved.
And remember, we put up a website this morning. What was it? Stemcell.org, I think. What is it? Stemcellfunding.org. And if you go to that website, it will put you in contact with your congressmen, your senators, draft letters for you, and very efficiently help you to get them off. So if that's what you believe, then you should definitely go that route as well.
Yeah.
Two more.
I understand that this could help grow myelin. Would that this technique do anything to help an atrophied spinal cord?
Well, it's an important issue. I think that the animal studies would support that dysmyelination is an important thing, even chronically, but that over time you tend to lose some axons that aren't being active, and the appropriate human studies just aren't possible. You need to use very detailed EM studies to demonstrate dysmyelination, and that's not possible in postmortem tissue. But the belief is that it would be still useful, years after an injury. Well, I think, as Jeff alluded, in our groups involved. Oh, I'm sorry. So when do you think that some strategies for remyelination will reach the human level? The answer is the phase 1 human studies are already beginning this year at multiple sites.
In fact, Jeff Coates had to leave. He's gone because he had to go. Where? Oh, Jeff's leaving soon then for human trials. So some of this for remyelination is coming very soon, but it will depend to a large degree on the cell, on what cell is being used.
Yeah. So right now, the human Schwann cells, as Jeff pointed out, are going to be entering phase 1 clinical trials for MS, and then we're doing a study looking at porcine stem cells for the purpose of remyelination, and two patients have already been completed in the phase 1. So phase 1 studies are only evaluating safety. So you need to go through the appropriate steps. The first step is phase 1 safety, and then phase 2 and 3 efficacy studies. So those take 5 to 10 years to complete, typically, that entire process of going through phase 1 through 3 trials. But it's so important that we begin now.